Hello, everyone. Welcome to our next session, which is on neuroinformatics and data sharing. We've got a pretty packed schedule. We've got 6 speakers. They're each going to give a 10-minute talk, followed by some questions. So please do get involved on the app, ask lots of questions. We also have the microphones if you'd like to ask them in person. My name's Amy, by the way.
I'm Nicola.
Fantastic. So yeah, if we can ask the speakers to try and keep roughly to time, just so we do have time for questions, that would be great. So let's get started. I'm very pleased to introduce our first speaker, Laura Waite, who is giving a talk titled, "A Data Steward Walks into a Bar, Experiences Managing Open and Restricted Brain Imaging Data." Thank you, Laura. Is Laura here? Okay, let's try again. Do the other speakers mind if we just go through, and then hopefully there'll be time at the end for Laura to come and give her talk. So we'll move on to the second speaker, who is Jacob Sands Robinson, who's going to give a talk titled "Continuous Testing of Neuroimaging Results Across Pipelines and Datasets." Thank you.
Okay, well, hi everyone. My name is Jacob Sands Robinson, and the abstract I submitted to OHBM this year is called "The Continuous Testing of Neuroimaging Results Across Pipelines and Datasets." So firstly, to give a little bit of context, right now there's an increasing amount of papers and studies that are coming out that make us call into question the rates of reproducibility of results in the neuroimaging field. And as it turns out, there are many methodological decisions that researchers can make that can affect the results that they get and lead to scientific result reproducibility issues. These can range all the way from the hardware that researchers choose to run their experiments on, to the different OSs and the different software environments they use, and even the very choice of pipelines and data sets that they use can impact the results. So specifically when we're focusing on the pipelines, we know that researchers are often faced with multiple choices of pipelines that have very similar capabilities. They can find the same things. And when you apply them to the same data, different pipelines to the same data, you can often get very different results. So there on the right, there's an image from a 2019 paper, and you can see that for the same brain scans, they run 3 different pipelines, AFNI, FSL, and SPM, and the brain scans, the brain activations in these all look slightly different, which is clearly slightly worrying from a reproducibility perspective. And this is worrying because it leads to a situation where there is no ground truth. If all of the pipelines give different results, how do you know which pipeline you should trust for your results? It's unclear which one to use, right? So another bit of context that I think is important to understand this presentation is that of continuous integration. So continuous integration has been proposed as a method to evaluate and facilitate the reproducibility of computational experiments. What exactly is continuous integration? It's a software engineering technique. It's very commonly used in industry, and we're starting to see its use in academia right now. And it basically consists of a series of tests that is launched automatically whenever new code is pushed to a code repository. So this is important because it allows you to check instantly whether the code that you've just pushed has the expected functionality, if it's doing the thing that you expect it to do. So while this has been done in a scientific context before, it is particularly challenging to do in a neuroimaging setting. This is because neuroimaging datasets tend to be rather large, and the pipelines tend to be rather memory hungry. So continuous integration platforms typically only really have a limited amount of memory that they offer people to run their experiments on. Another limiting factor is the fact that datasets, medical datasets, often have very heavy privacy conditions, which make it hard to just put your data in a public repository and run tests on it. So to solve these limitations, we built an infrastructure, a continuous integration-based infrastructure, that is able to run many pipelines on many data sets for one single experiment to evaluate the variability of the result in this multiverse of conditions and methodological choices. And it offloads all of the really heavy computations and storage to a distributed system. So what is the goal of this presentation? The goal of this presentation is to use this continuous integration framework that we built to investigate a neuroimaging use case. So we're going to be investigating the associations between hearing loss and brain structure, which is a particularly interesting result in the field because its robustness and reliability is unclear. It changes depending on the pipeline and the association measure that researchers choose to use. So while some studies agree that hearing loss can reliably predict gray and white matter atrophy and is strongly associated with dementia, there are also other papers that have null results. They find no association between hearing loss and brain structure. And it's also important to consider publication bias. In academia, there is a strong bias for significant results to be published over null results. So many studies with null results were probably simply never published. This is known as the file drawer effect and has to be taken into account. So in terms of a method, I don't have time to go into a ton of detail, but very quickly I will explain the 4 main modules of this continuous integration framework. It was important to make it modular so that you can add new pipelines and data sets on the fly continuously, because in neuroimaging, new tools and new versions of tools and new versions of data sets and new data sets are constantly being pushed, so to speak. They're constantly being published. So the first module is the data retrieval module. And it consists of a script which basically allows you to pull distributed data sets from a dataset repository. In my case, I'm using the Canadian Open Neuroscience Platform's repository. And it pulls them onto the distributed server of your choice, in my case, the Compute Canada servers, using a piece of software called DataLad. The second module is the pipeline containerization module. And it allows us to containerize all of the pipelines that we wish to use to run our experiments with. And we create descriptors for them that allow them to be portably executed on these distributed servers. And we do this with a piece of software called Boutiques. So at this point, we have our pipelines and we have our data sets. And then we wish to hook them up to our distributed server. And we're doing this with a distributed computation, a cloud computation platform for neuroscience called CBrain. And the reason why CBrain is so important, and as this intermediate layer in between the distributed servers and our other modules, is that it features an API. So we can control we can orchestrate and control all of the distributed computations from a script or from a local or a remote place. And then the fourth module, and really the steak and potatoes of the meal, the central one, so to speak, is the continuous integration module. And CBrain is precisely important because it allows us to execute distributed computations using its API in the continuous integration setting. So when someone goes on GitHub and forks this repository, they will find a user input file called the experiment definition file. And there they can provide all of the IDs in there for all of the pipelines and data sets they wish to execute their experiment on. So then they'll edit this file, they'll push it to GitHub, and then this will trigger a continuous integration run, the automated tests. We're using Circle CI, a free continuous integration client that hasn't crashed too much so far, so that's pretty good. And basically then what happens now is that while Circle CI is running, it does all of the newest computations that people can specify and in their experiment definition file. And at the end of every run, it fetches all of the latest results that have been computed, it processes them, and then it creates interactive visualizations and statistical summaries of all of the newest computations so far. So in terms of results, what have we got? Well, firstly, we have the fact that, the continuous integration code, this framework's code, NeuroCI we've decided to call it, it's on GitHub, so you can all have a look at it, you can all fork it, and hopefully you'll use it and let me know if anything bugs out or if it works nicely. And then what we did was we decided to use the PreventAD longitudinal Alzheimer's data set to evaluate left hippocampal volumes as just one example measure of brain structure and compare it to dichotic stimulus identification scores, which is a hearing loss measure. Typically, a higher DSI score, dichotic stimulus identification, means better central auditory system function. So we calculated these left hippocampal volumes for now on two pipelines, two very popular pipelines, FSL and FreeSurfer. And as you can see from the two graphs there, they don't look quite the same. And if we look more in depth as to what's going on, for example, if you look at the XY plot on the right, you can see FreeSurfer volumes on the left, on the Y-axis, FSL volumes on the x-axis, and you can see there's a pretty big discrepancy. In fact, the Pearson correlation between the two pipelines after outlier removal is only 0.2. And by outlier removal, I mean results that were outside of a biological plausible range. So if we look at the distributions on the left, we see that the two pipelines output quite different looking distributions. FSL has a larger spread in values, an asymmetrical distribution. It's skewed towards the lower volumes. So that's the one in blue. The one in red is FreeSurfer, and the distributions are roughly symmetrical, and they consist of higher values. This higher value of volumetric measure output by FreeSurfer over FSL is also documented in past literature. So then, I'll move on to the conclusions, keep this short and snappy. We've created a framework which can iterate many pipelines on many data sets for a given experiment. And this allows us to not only systematically evaluate result variability in experiments, but it also can allow users to pinpoint biases and discrepancies caused by certain tools and data sets, and actually quantify their impact on the results. And this is very important. It's not just for my own use case of hearing loss associations with brain structure. It can generalize, the tool can generalize to many other neuroscience and neuroimaging domains that researchers may wish to investigate. And yeah, it's very important because it allows us to actually consolidate knowledge in the field and explain uncertainty in fields where there are reproducibility concerns, such as neuroimaging right now. For health-related use cases, such as my own, having these reliable results is really important because it can allow both researchers and practitioners to move towards the most likely robust biomarkers for their research and practice. And in terms of future work, what I'm going to do, seeing how discrepant the pipelines were, I wish to investigate this problem a bit more, and so I'll add many other pipelines that allow me to compute left hippocampal volumes, such as civet, ants, mink tools, probably ashes, probably SPM, AFNI, just throw the kitchen sink at them. And then for the data sets, I will choose a couple of more data sets to add, which will allow me to investigate the hearing loss use case a bit better, such as Compass ND and the UK Biobank. And that's all.
Don't run away, you haven't finished yet. We've got some questions for you. Very nice talk, by the way. So some people want to know, how do you deal with the long-running pipelines and the fact that some pipelines will take different timelines? That's one of the questions. Yeah, so for many of these neuroimaging tools, the pipelines take a very long time to run. FreeSurfer can often, on the distributed system, take over 8 hours. I find continuous integration services typically have a cron job option, which means that you can repeat the continuous integration run after a push every X amount of hours or something like that. So every time the continuous integration service runs, it is pulling the newest set of computations and the current progress from the distributed system. So it basically updates its cache files where it's storing all of the intermediate steps of the computation and all of the task statuses. It can pull them at the beginning of each run from the distributed system. So it keeps track of that automatically. And the next time there's a continuous integration run, either because you changed the files or because you just ran it on a cron job, it will automatically deal with this.
Excellent. One more very quick question. How do you prevent users from mining crypto on Seabed?
Yeah, that's a good one. Basically, to use, when you integrate a container with a pipeline into boutiques, and then you send this container to the CBrain administrators for integration, they check that the descriptor of the container hasn't got anything too fishy going on. Excellent. I've been told off before by the administrators of it for not having appropriate security on these containers. So that was definitely a learning experience, but thankfully no Bitcoin were mined.
Okay. Jacob, there are many more questions, but because of time constraints, could you just look into the app? Do you have the app?
Yes.
Okay, we'll look into the app and you can answer them afterwards yourself, because unfortunately we don't have time to answer all the questions right now, okay?
Okay, that sounds good. Check out NeuroCI on GitHub. Cheers.
Fantastic. Excellent. So our next talk will be by Anais Artigas, and the talk is titled Ginkgo, a novel modular and open source MRI pulse sequence development framework.
Hello everybody, I'm Anais Artigas, and it is my great pleasure today to present you a novel modular and open source MRI pulse sequence development framework that we named Ginkgo. As many of you may know, when you want to develop an MRI pulse sequence, you will need a development kit from your MRI manufacturer. And this can come with some issues regarding intellectual property or access to some codes that may be hidden or just in the form of binaries. And sometimes it can be very difficult to get into the code that is very well embedded in the manufacturer's framework. So we thought that an open source project will provide accessibility and a participative environment for MRI pulse sequence development. And we would like very much it to be cross-manufacturer, but for now we developed it only for Siemens. Our big example was the Gadgetron framework that is meant to reconstruct the MRI images in an open source framework. So here is a class diagram of the Ginkgo framework. We built it in an open source system that is based on factories that are generating sequence modules. Those modules provide a place in the timeline for any part of the sequence. They will give an anchor time and a duration for any module building the sequence. Deriving from this sequence module, you will find preparation, excitation, refocusing, readout, and spoiling modules categories that are covering all the different modules you need to build any MRI pulse sequence. Deriving from them, you will find very specific modules needed to build the sequences that you want. For instance, here we built a spin-echo 3D EPI sequence using the framework. To do so, you may want to have some preparation modules like fat saturation or diffusion weighted, but we did not include them in here for now. You will just have the core modules of the spin-echo 3D EPI sequence, which are the single TX excitation module in blue, including the radiofrequency excitation pulse and the slice selection gradient, the refocusing 3D module, including the refocusing pulse, the slice selection, and the crushers, the ecoplanar reading 3D module, including the ecoplanar train reading gradients, and the spoiling module with the spoiling gradients. Here is a bit of code. So we used the main structure from the Siemens framework, which got 4 main methods: the initialize method, getting the parameters of the sequence; the prepare method, setting the events of the sequence, like the gradients, the radiofrequency pulses, the ADC converter events; and the run kernel method, setting all those events in order in one time kernel, which usually is 1 repetition time; and the run method looping over the lines, the slices, the partitions, the repetition times, even the diffusion orientations. If we look at figure 1, you will find an extract from the constructor of the spin-echo 3D sequence. So you can see here that we use the sequence module factory to build the different modules that we need. You will find the excitation module in the first place, which will be the first in the timeline, and then all the different modules that you need in the time order. Sometimes you will need to set some parameters, like for the excitation module, here you set the flip angle and the parameters for your radiofrequency pulse. After all the modules are set, You will only need to set their timing in the repetition time in the run kernel method as shown on figure 2. So you will set the anchor times, which are meant to be easy to place on the timeline. And after that, the code will run all the inner methods of the modules by itself. The only thing you will need to do after that is to set on where you want to loop. So this will go in the run method, and for instance here, as it is a 3D sequence, you will need to loop over the lines and the partitions. So how do you implement the framework? So we hope this has been made very simple. So we have a Git repository. You will find a QR code pointing on it on my last slide. You will need to download the codes from it and to put them in your IDEA-sec file. And then you must be able to compile directly the already released sequences. And you can also modify the code wherever you want and compile it with your own sequences. Then you will put the compiled file on the console and acquire your images. To reconstruct the images, you have two options, at least for the basic sequences. You can use the Siemens reconstructor, which is ICE, but we hope that you will also be able to use the gadget run to reconstruct the images because it is also open source and allows more complex reconstruction and more flexibility in the sequence. We try to have a gadget run reconstructor for any of our sequences so that you can use them with the gadget run. It is to be noted that the gadget run also provides online reconstruction if you need it. Here are basic results we obtain with the framework. So we choose to test it on the very simple sequences, which are gradient echo and spin echo, 2D and 3D. We did ex vivo images on a postmortem Nyala brain. So for the small story, a Nyala is a kind of antelope. So this is what you see here. And if you look at the figures, you will see that you have a timeline. And this timeline shows the different modules that you need to implement to have this sequence. Some modules are in common. This is the whole aim of the framework, that you can use different sequences with the same modules if you need to. We also wanted to try a more elaborated sequence, so our choice pointed to the spin echo 3D EPI sequence. So I must have a disclaimer on these images because this sequence has still to be accelerated with the partial Fourier and the graph algorithm so that we can get better results. But still, you can see that the image seems to be pretty good. And we also weighted this sequence in diffusion and obtained the fractional anisotropy map that you can see here, showing the main tracks of the brain. We wanted to have the full access to the sequence code so that anybody can modify everything everywhere in the code and get inspiration too. And we wanted to have an object-oriented architecture that is really clear with clear variable names so that the people can just read the code and get inside easily. And we had to choose a first device, which was the Siemens device we had at the lab. And we tested everything on IDEA V11C, but it should be easy to implement it on later versions with minor changes. And using this toolkit, we built a hierarchical decomposition of any MRI pulse sequence. And everything is compatible with the reconstruction in an open source framework named GadgetRun. And it is available online on our Framagit repository. So we managed to build an open source modular MRI pulse sequence framework development framework compatible with Siemens for now, and we verified its pertinence on postmortem and phantom images, and we provided a clear architecture that is easy to read for the user, and for now it is enabling basic diffusion and rapid imaging sequences. As future work, we would like to implement it for other vendors like GE, Philips, and Canon, and ideally provide a code that is cross-manufacturer, that you can just program the sequence and then translate it for your choice manufacturer. And we would like to cover a broad range of diffusion MRI schemes and test them on human volunteers. I will soon provide an online documentation so that it is easier to implement it, and any contribution from the community will be very, very welcome. I would like to thank my supervisors, Cyril, Philippe, and Ivy, and all my team for helping me in this work. Thank you for your attention. Do you have any questions?
Very nice talk. There are some questions that you've actually answered already, like people wanting to know if it would be compatible with other providers and other MR vendors. So that is in your future developments.
Yeah.
Are you also not just thinking about you providing the resources to be able to apply it to other vendors, but are you contacting these other vendors to ensure their support?
Actually, I haven't, because this is a good idea, but the project is not started with other vendors, so I can assure you that it will be possible. But I think as long as it is open source and we don't owe anything to the vendors, this is our own code, so it should be possible.
And one more question, how is the integration to the scanner working?
So it is very simple. It is like you wanted to have a sequence from Siemens, and they gave you the sequence, and you have the compiled file, like the DLL and the ASO files, and you just put them in your customer sec folder in the scanner, and then you can run the sequence. It is exactly the same. You just need to compile the sequences on your own IDEA version, and once it's compiled, you can just put the files into your console's customer sec file, and then the sequence should work and you will get the images. Maybe the difficult point is when it comes to reconstruction. If you're not enabling the ICE reconstructor, you will need to retrieve the raw data or to implement an online reconstruction using Gadgetron, which can be a bit tricky.
Thank you. Again, because of time constraint, we're going to have to cut the discussion here, but there are more questions in the app. So if you'd look, do you have the app installed?
Yeah.
Right, wonderful. We'll have a look at it then.
Thank you.
Excellent. So our next speaker will be Sebastian Bludau with a talk titled Layer-Specific Cortical Cell Distributions of Cytoarchitectonic Areas Anchored in Big Brain.
Okay. Hi, everybody, and thanks for the opportunity. Do I have to wear the mask?
Go closer to the microphone so we can hear you.
Okay. The topic of my talk is layer-specific cortical cell distributions of cytoarchitectonic areas anchored in big brain space. My name is Sebastian Bludau, and the project was developed together with my colleagues from the Research Center Jülich under the supervision of Professor Amons. So, brain simulation, brain-inspired AI, and other neuroscientific disciplines are using microstructural measurements like regional cell densities to model differences between areas in their computation and simulations. In order to provide reproducible and precise cell numbers for the analysis just described, we have decided to use the big brain to extract the data sets. The big brain itself is a brain which was sectioned into 7,400 histological sections. Those sections were stained for cell bodies, and they were digitized with a resolution of 20 micrometer in plane. And because of the section thickness of 20 micrometer, the big brain model itself has an isotropic resolution of 20 micrometer, which already allows for anatomical analysis. But it was the goal of the here presented study to increase the resolution in selected areas to 1 micrometer to even allow for single cell segmentations. Therefore, we have extracted small image patches with a size of a few square millimeter in 12 different areas of the Jülich Brain Atlas. The Jülich Brain Atlas is an atlas based on microstructural differences of areas, and it's freely available, and it consists of more than 150 maps which are included in this atlas, and for all those maps there are individual peer-reviewed studies available. And additionally, this atlas is also deeply embedded into the eBrains infrastructure, which does, on the one hand, mean that you can easily switch between different reference spaces, like the Big Brain reference space or the MNI 152 reference space, but you can also enrich the data with different modalities which might be available for the different areas, like receptor densities or connectivity data. So here we can see one example area where we've extracted such patches. It's area FP1, located at the frontal pole of the human brain. And within those maps, which are probabilistic, which mean we have analyzed 10 different brains to create such an area to also address the inter-individual variability. And we have extracted the image patches at position with a very high probability of being the specific area. And additionally, we have extracted the positions at parts of the tissue where the surface normal is approximately parallel to the original cutting direction of the big brain, in order to fulfill the requirement that the tissue was cut perpendicular to show a clear lamination. At one last pre-processing step, we had to transfer those coordinates to the big brain space. And here we can see all 120 patches, the position of those patches which were extracted for the presented study. And by plotting them in their spatial relationship, you can directly analyze them according to the neighbor. But you can also enrich the patch positions with information which is already available in the big brain model itself. So we can directly benefit from the high degree of information which is available there. So for example, we can extract laminar-wise gray value intensities, which we also have done here in this study. I will present it later. And we are still collecting more data. So just in a second, we see a few more blue spheres. Those are patch positions which have already been extracted. And we are currently analyzing those patch positions and the patches themselves to include them in the project in the very near future. So the redigitized images of the histological sections were digitized with a resolution of 1 micrometer in plane, which allows a clear detection of individual cells which have approximate size of 5 to 20 micrometer. Afterwards, we have applied state-of-the-art deep learning algorithms to segment the individual cells. And this results in area and laminar-specific cell densities. And furthermore, the algorithm also allows for biological, meaningful, and reproducible splitting of touching cells. But we also included the raw data. So if someone wants to use his own scripts or methods to segment the images, you can do it, of course, and you can verify the detection of each individual cell. So the patch positions, the images of the just described positions were subsequently annotated, manually annotated for the cortical laminas from 1 to 6. And those annotations were verified by trained experts. And here we can see 2 example patches, one for the primary motor cortex and on the right side for the Broca area. And altogether, it's a data set of about 6 gigabytes. And the complete data set itself is also integrated into the eBrains infrastructure, which does mean on the one hand that it's really enriched with metadata describing all methods, reference spaces which were used to create the data set, and all data sets do have a citable DOI so that you can directly use it if you want to. So analyzing the area and laminar-specific cell densities showed clear differences, of course, between the different areas. So, and classical cytotectonic differences, like differences between the different layers, could be reproduced over the given sample size. And since it's embedded in the eBrains infrastructure, you can use the Zebra Python library, the eBrains Zebra Python library, which allows you programmatic access to all the data sets. So there are examples available, notebook examples, which can directly be executed in notebooks. And it's very easy to get those data and incorporate them into your own workflows and scripts. And the next step, we try to create interpolated cell densities to receive whole brain coverage. So therefore, we have extracted gray value intensities in the specific layers of the areas we have analyzed. Therefore, we have used the already provided layer annotations from the Wechsel publication. And we have correlated those gray value intensities with the 1 micrometer measurements we have made here in our study. And we have created a kind of calibration function using those 2 measurements. And by applying those calibration functions to all areas which are included in the Jülich Brain Atlas, we could create layer-specific cell densities for all areas included in this atlas, which here on the left side you can see those cell densities plotted on the FS average template. And we can see here a trend of decreasing cell distributions along the posterior-anterior axis, which is more pronounced in the granular layers, layer 2 and 4, than in the pyramidal layers 3, 5, and 6. On the right side, the rotating plot shows the cortical mean cell distribution, so the mean over all 6 cortical layers plotted against their X and Y position of the corresponding Jülich brain area, which again shows this decreasing cell density along the posterior-anterior axis, but it also shows the large degree of divergency along this trend, which underlines the importance of precise area and layer-specific cell densities here. Additionally, we have found quite large differences in some areas which are sometimes treated as one large area. So in some atlases, for example, the insula is treated as one large area, or at least it's only addressed by one label. The Jülich Brain Atlas have shown that there are microstructural distinct areas in this area. So it's divided in up to 16 distinct cytotonic areas. And if we plot the layer-specific cell densities for all those 16 areas, we can see a variance of up to 15% in those different small areas, which again, on the one hand, shows that we can reproduce this finer parcellation, but also that such a fine-grained parcellation is necessary to extract really the full degree of information and complexity of such a large region like the insula. So, coming to an end, I would like to conclude my talk. So I've just presented you a data set containing high-resolution histological images with manually verified laminas, which were analyzed for their cell densities, and the complete data set and project is embedded in the eBrains infrastructure. The results are reproducible, so the segmentation of each individual cell can be reproduced. You can apply your own scripts if you want to work with your own scripts. All patches are spatially anchored in the big brain space, which allows you also easy transfer to different reference spaces. And this transfer can, for example, easily be done using the Zebra Python library, because it's integrated into the multi-atlas framework, so which allows you an easy access, but also this allows you to enrich the data with the different modalities which are also available in the eBrains infrastructure. And of course, I would like to thank my colleagues, especially Timo Dickscheid, who drove forward the methodological developments, and Katrin Amunds, who made the study possible. And I would like to invite you to my poster 710 tomorrow, sometimes 12:45. Thanks a lot.
Wonderful talk, Sebastian. Thank you. We've got questions as well. So there's somebody asking, scanning images, whole human brain sections in 1 micrometer resolution. This poses big problems concerning time and also amount of data that's produced. Exactly how long do you need, and exactly how much data do you produce?
Yes, it's definitely very time consuming. We are in the lucky position in our institute that we have quite a lot large microscopes which are automized so that we can place up to 10 sections in one scanner, and this microscope starts working for about 4 days automatically. But scanning one whole brain section takes up to 15 hours. Depends a bit on the size of the sections. And if we are collecting also a focus stack of the complete image. Normally we have a data size of approximate 8 to 10 gigabyte per image and per focus level. So the amount of data is also quite high. But yeah, therefore we have 10 of those instruments right now in Institute to have a kind of parallelization here and to keep forward.
And how many sections, just like to scale it up, how many sections does the big brain have? You know, because if you're talking about so many hours for one section, and I know that the section size has changed, but
so the amount of instruments we have in Jülich are designed to be able to scan one complete brain in about 1 year, if everything works fine. So of course that's not always the case, but it's approximately 1 year per brain. The big brain was published in 2020. This was digitized with an in-plane resolution of 20 micrometer. So there the amount of data was very large at this time, but nowadays it's quite handy. It's about 1 terabyte. It's quite easy to handle, but this was of course faster.
A very manageable amount of data. Impressive. Also, I've got a personal question, if I may ask, because there are more in the app, but I'd like to squeeze this one in. You've got not just like, you know, global cell counts, but you've also got laminar information. So for me as an anatomist, this is important. But why, you know, why did you decide to move down? Because this is an even larger effort, obviously.
Yeah, definitely. But we think it's worth all the work, because on the one hand we have seen on the slide that we have different trends of decreasing cell densities throughout the layer, so they do not all behave the same. And we know that all the layers are differentially connected to each other, so if we really want to extract the full degree of information, we should use layer-wise and area-wise precise numbers. And you can still merge them to create a mean value. That's still possible, but I think it's definitely worth to go into the detail here.
So again, because of time constraints, check the app.
I will have a look in the app.
Thank you really very much. Pleasure. So it's my pleasure now to introduce Amy's talk. She's going to talk about the Big Mac data set, linking tissue microstructure with MR signals throughout the macaque brain.
Fantastic. Thank you so much. So yeah, my name's Amy Howard, and today I'm going to be talking to you about our recent work on the Big Mac dataset. So our ability to characterize the connectome has been greatly advanced by the scientific community's access to big data. But I would say there's different ways in which data can be big. First of all, we can look across populations for between subject variability, such as within the 1,200 participants of the Human Connectome Project. Alternatively, big data can act to characterize a single brain in exquisite detail. And a beautiful example of this is the My Connectome project, where a single subject was scanned 3 times a week for over 50 weeks. Recent work in the macaque by Ortio devises an HCP-style macaque protocol, which not only will improve the data consistency across multiple subjects, but also provides an important link to the human HCP data for cross-species analysis. But to characterize a single macaque brain in exquisite detail, we present the Big Macaque or Big Mac dataset. The Big Mac dataset includes in vivo MRI, post-mortem MRI, and multi-contrast microscopy in a single whole macaque brain, where upon publication, all the data and tools will be made open access. The in vivo data was acquired over multiple scan sessions. We have around 270 hours of post-mortem MRI and thousands of hours of microscopy, which results in several terabytes of data. So let's unpack this a little. So the in vivo data includes structural MRI, task fMRI, resting state, and diffusion data, whilst the post-mortem MRI includes structural T1 maps and an extensive diffusion protocol I'll come to in a second. But having both the in vivo and post-mortem data really provides a fantastic opportunity for us to link our post-mortem results to in vivo data within the same single brain. The post-mortem diffusion protocol includes 2 spatial resolutions. So at 0.6 millimeters, we have a p-value of 4,000, and at 1 millimeter, we have p-values of 4, 7, and 10,000. The high spatial resolution data allows us to characterize single fiber populations with fewer partial volume effects, whilst the 1 millimeter data allows us to obtain good signal-to-noise ratio, whilst we push to higher b-values, and also we can achieve 1,000 gradient directions in the outer 2 shells within a reasonable scan time. Further, at 1 millimeter, we have both linear and spherical tensor-encoded data for more advanced microstructural modeling. And of course, at high b-value, we obtain very good diffusion contrast. So here I'm just showing different diffusion-weighted images as we trace out this spiral on the Q-space sphere. So all of our data was acquired on a 7 Tesla small animal scanner using a spin echo multi-slice sequence. So this sequence is really good because it has very few imaging artifacts, but it is fairly slow, which is why we had to scan for over a week. So we want to relate our MRI signals to microscopy data, which has both ultra-high spatial resolution, but also high specificity. So what microscopy data do we include in Big Mac? After scanning, the whole brain was sectioned along the anterior-posterior axis, where we collected consecutive slides for either polarized light imaging or one of 4 histological stains. And each contrast is repeated every 350 microns throughout the brain, and we have whole brain coverage. Here's some example microscopy data that we have acquired so far. So on the left, I'm showing polarized light imaging, which utilizes the birefringence of the myelin to estimate the primary fiber orientation per pixel at a resolution of around 4 microns. And then we have 2 different histological stains. So in the middle, we have a myelin stain where the myelin has been stained brown. And on the right, we have crescent violet staining of Nissl bodies, where we can see the cell bodies in great detail. And all of the histology is acquired with submicron resolution. We can then perform structure tensor analysis on the histological stains to extract the primary fiber orientation per pixel. And here I'm showing the output from the myelin stain. However, we're not limited to performing structure tensor analysis only on myelin. On the left here, I'm showing what happens when we apply structure tensor analysis to the Nissl stain slides, where we can see really good orientation information because the cell bodies tend to align with the orientation of the fibers, as shown in the bottom. We can see that there's very complementary information from both of these stains, where both of them are even able to distinguish these intermingling fiber populations within the corpus callosum. In Big Mac, MRI and microscopy data has been co-registered using TURL, which has been recently developed by another member of the group. TURL is a very powerful platform that allows us to map every 2D microscopy pixel into a 3D MR volume. Here I'm showing some example results where I've registered the structural MRI to the PLI data on top. And you can see that in the structural MRI, there are kind of green boundaries that are showing the white-gray meta boundary, and also orange lines which show the tissue tissue edge. And when I overlay these lines onto the PLI slide, you can see that we do get very good correspondence. This means that we can have meaningful voxel-wise comparisons of the MRI and microscopy data in Big Mac. So we believe there are a wide variety of applications for Big Mac, including neuroanatomical investigations, multi-scale neuroscience, MRI methodology and validation, particularly with respect to diffusion models, as well as machine learning and data fusion. But today, I'd like to just provide one example analysis of what we could do with Big Mac. And this example is really based on the recent acquisition of these Nissl stain slides that allow us to visualize the cytoarchitecture in great detail. So of course, we can see the cortical layers with the different cell densities, but we also have this orientation information, as I explained earlier, where the cell bodies tend to align with the fibers. So our question here was really, can we link this microscopy-defined microstructure with MRI signals throughout the cortex? Our method is inspired by some recent work in Big Brain, where we extract these cortical profiles, which really utilize the natural organization of the cortex. And a cortical profile is just a 1D profile which extends from the white-gray to matter surface to the peel boundary. And this is a really nice analysis framework because not only do these profiles provide a really rich description of the data, but they also provide a common access for meaningful MRI and microscopy comparisons. In Big Mac, we used an adapted free-surfer pipeline to extract these cortical profiles. So we extract a series of surfaces across the cortex, and these surfaces all have matched vertices, which means we have a series of 1D profiles which then are across the entire brain. We then needed to map these cortical profiles onto our 2D microscopy data, and this was again achieved using TURL. So on the left here, I'm showing some example preliminary results where we have mapped these cortical profiles from the 3D domain onto the 2D microscopy slide. We can then sample various modalities along these profiles, where here I'm just showing the average profile from this region of interest. And in today's results, we analyzed only the left hemisphere with over 280,000 profiles per contrast. The postmortem MRI metrics include the structural intensity from a multigradient echo, as well as fractional anisotropy and mean diffusivity from diffusion tensor analysis of B4000 data. Our microscopy metrics include the Nissl density, which is just a proxy for cell body density, as well as Nissl dispersion, which describes how coherently oriented the microstructure is, and this was an output from the structure tensor analysis. So in our first results, we did a very simple correlation. So we just take the average profile from each of these regions of the CHARM3 atlas, we correlate the profiles on a region of interest basis, and these histograms show the correlation coefficients across regions. The red line indicates the mean. So as we can see that we have a really nice correspondence between the MRI, structural MRI and the Nissl density and Nissl dispersion, which correlates negatively with the density and positively with the dispersion, and that these are fairly consistent results across the different regions. In comparison, the diffusion tensor metrics show a much less clear result or pattern of results, with some regions correlating very strongly, positively, and some negatively. And so I think this really demonstrates the pitfalls in current region of interest validation studies, where you may come to very different conclusions if you only considered two different regions of interest. Instead of just correlating these different modalities, can we do something more and combine them into some sort of joint analysis? So here I'm showing the results from running canonical correlation analysis, which allows us to find common modes of cortical variation. And here I'm just showing the correlation coefficient of the first component. As we found, we got a fairly nice correlation between the Nissl density and the structural MRI, and this is reassuring as it mirrors recent results found in humans. And then, of course, we can take this correlation axis and we can map it back onto our CHARM3 atlas, where in these preliminary results we have this nice separation of the frontal, occipital, and the temporal parietal areas. In comparison, we see only a very weak correlation with the diffusion metrics, and this suggests that really we see structural sensitivity to brain-wide patterns of cytoarchitecture, as described by the Nissl density, and that's true with the structural MRI, but not with our diffusion metrics. So to conclude, today I've introduced the Big Mac dataset, which is a new resource combining in vivo MRI, postmortem MRI, and multi-contrast microscopy in a single whole macaque brain, where upon publication, all our data and tools will be made open access. Further, I've demonstrated one example analysis with a pipeline to extract these cortical profiles, where we found that structural MRI, but not the DTI metrics, shows sensitivity to brain-wide variations in the Nissl cytoarchitecture. Future work, we're going to include even more of the contrast that we have within Big Mac into these multimodal comparisons. So that'll include more microstructurally specific diffusion metrics, as well as myelin-stained histology and also the in vivo signals. With that, I'd like to thank everyone who's been involved, and thank you very much for listening.
Wow, another nice talk. We've only got time for one question, though. And I've been asked, or you've been asked, basically, is it practical to train and distribute a model to infer enhancements to in vivo macaque MRI using the matched in vivo ex vivo pairs?
Yes, I think this is a really exciting avenue. I think, in postmortem imaging, often we really lack the translation of our postmortem results to in vivo data. And I think having both modalities within the same animal really allows us to explore those effects. And as ever with postmortem data, you really want to have translation and impact on our in vivo methods. So yeah, it's a very exciting opportunity. Thank you. Okay, so our next speaker is Julie Boyle on the Courtois Neuromod project, a deep multi-domain fMRI dataset to build individual brain models.
I don't know if everybody could hear me. This podium's not made for little people. I can only see part of the audience. So my name is Julie, and I'm here today to talk to you about the Courtois Neuromod project. So it's a deep multi-domain fMRI data set to build individual brain models. So one of the main objectives of the Courtois Neuromod project is create a deep neuroimaging data sets that probe numerous cognitive domains, including vision, language, audition, emotions, memories, and video games. And then the plan is to use those data sets to train artificial neural networks on individual brain activity and behavior. And the reason I'm here today is actually to talk to you about this last point, which is we would like to then take those data sets and share it with the community. So I'm going to talk to you a little bit about the data that we have and how you can get access to it if you would like it. So first, before I delve into the data, I just want to define the meaning of the word deep. So at Neuromod, when we speak about deep, we mean deep individual sampling. There's this really beautiful graph by Nasalaris et al. That was published in 2001 that really does a great job of defining the meaning of deep, at least in Neuromod. So what they've done is they've graphed various data sets available to the community on two axes. On the y-axis, you have the number of subjects that they scan in those data sets, and on the x-axis, you have the hours of fMRI per data. And what you see is in the upper left-hand side is a UK biobank where they've scanned thousands of people for a few hours each. And in the bottom left-hand corner, you see the natural scenes data set where they've intensively scanned 8 subjects for 40 hours while looking at naturalistic stimuli. So where does Neuromod fit on that graph? Well, we're a little bit off of the x-axis, and I think in part that's probably because they actually had to publish this in a scientific paper, so there's sizing restrictions. So what I'm going to do is extend the y-axis to let you know about where we are at with the data that we have when we submitted this OHBM abstract. So if we were actually put at least the x-axis to scale, apologies we did not put the y-axis to scale, Neuromod would be somewhere here. So we currently, at least when we submitted this poster, we had 100 hours of fMRI data per subject, or more or less. I'll tell you more about that in a second. And to give you an idea of where we're heading and where we want to bring the project, by the end of 2024, we're going to reach about 350 hours of functional neuroimaging data, which will represent about 500 hours of fMRI and MEG acquisition time. So I'm going to tell you about the kind of people that would agree to be scanned this intensively. So you can imagine our inclusion criteria for our participants were not too stringent. People needed to be just generally healthy. They needed to be MRI and MEG compatible. They needed to have a normal hearing for their age. They needed to have a solid comprehension of the English language, and that's because all of our stimuli are in English. And last and most importantly, they needed to be willing to be scanned for 1.5 to 3 hours a week for at least 5 years. Yeah, you read that correctly. So whom are these individuals who would agree to do such a crazy task? So these are our 6 subjects. We happen to have recruited 3 women, 3 men, fluke. At age of recruitment, they vary between 31 and 47. All participants were right-handed, also another fluke. And 3 of the participants reported their maternal languages being French, 1 as English, and 2 reported their maternal languages as being bilingual. So again, before I talk about the data, I'm going to give you an idea of our scanning setup. What you see on the right-hand side is what we do in the MRI scanner. On the left-hand side is the MEG, and what you find in the middle is stuff that we mirror on both sides, both in the MRI and the MEG. So today's talk will really focus more on the MRI, because that's the data that we're prepping to, that's being released, at least as a first release. So we use a 3T PrismaFit scanner. Participants hear stimuli using Sensometrics S15 headphones and custom-built ear protection. You can imagine when you're scanning someone that much, you want to make sure that you protect their ears. We use an MRC eye tracker. Participants wear custom CaseForge head cases. And while they're being scanned, we use a biopack system, and we acquire signal on their ECG, their pulse, their respiration, and their skin conductance. And anything that requires any type of action or response, we use our custom-built video game controller. So this is our data set that's as of December 2021. So what you can see is that in at least 3 of our subjects, we had just crossed the 100-hour mark, with a 3rd subject just slightly behind. Since then, these subjects have all passed the 150-hour mark. What you'll also notice that 2 of our participants are less intensively scanned, and this a little bit has to do with life. When you're doing such a longitudinal study, you can't guarantee that everyone's going to be as available, and with COVID, we definitely had 2 participants that were less available. So the plan moving forward is to have 4 hyper-scanned or intensively scanned participants and 2 that are less scanned. But the 2 less scanned will still represent about 40 to 50 hours of fMRI data per year, so it's still a chunk of data. So what have we acquired so far? You'll see that we've acquired a lot of Friends. We've acquired season 1 through 5 of Friends. We've done the HCP tasks. So we've done HCP TRT, where we repeat the HCP tasks 15 times. We've done video games. We're big fans of video games at Neuromod. So we've done Mario and Shinobi. We've also done a variety of visual localizers, and we've done a data set we're calling Things, where participants did a 36-week memory task using the Martin Hebert's Things images. So if you'd like more information about these data sets, just go to docs.cneuromod.ca or drop by the poster, ask me about it. I'm always happy to talk about Neuromod. So just to give you an idea of what we're planning to acquire and what we've started to acquire in 2022, 2023, the type of stimuli that we're acquiring really divide across 3 types of tasks. One is a scanner validation setup. We have a data set called Audition, where we're going to do auditory thresholds in the scanner to be able to assess the sound quality that our participants are experiencing. We have a lot more cognitive tasks coming, so if you're interested in those, there's more coming. We have Image 10K, which is 10,000 images that are available in the public domain, so they'll be publicly available. We have a triplets task. We have an emotions task, which is based on the Cohen gifts that we'll be acquiring. We have a lot more localizers, language localizers and vision localizers that we're currently acquiring, and a task called multi-FS, where it's multitask, multi-feature, multi-stimulus. In terms of naturalistic naturalistic tasks, there's more video games. We're currently acquiring Mario Stars, and we're hoping to acquire Mario Meg if the Meg manages to reopen. And Mario 3 is the next thing in the scanner. We're also going to acquire narratives data set this year, and season 6 and 7 of Friends. So if you're interested in video stimuli, this is the data set to check out. So how do you access the data? I think that's probably the most important part of this talk. I'm really glad to announce that 4 years after we've started this project, this data set, or the data bank, is finally open. And if you want to hear a really long story about how to open and share data over a beer, stop by my poster and I'll tell you the 4-year journey of how we were able to share that data. So in order to access the data, you need to be a principal investigator with university credentials. You need to have, just write a really short blurb about what you want to do. It doesn't need to be super specific. We just want to make sure that it's congruent with what we've consented our participants for. And our institution needs to sign data transfer agreements, and then the data's yours. The data are in BIS, they're structured in DataLab, and we've pre-processed some of the all the data with the fMRI Prep LTS. So if you go to the cneuromod.ca website, there's a big green button on the front page. If you click on it, you can get access to it, and it's open and functional as of Thursday. So in conclusion, by the end of 2022, we'll have acquired 8 new data sets totaling up to 200 hours of fMRI and imaging per subject, at least in those 4 intensively scanned subjects. I think this will make us the largest functional neuroimaging database collected on individual subjects. And what we hope is it opens a unique avenue to build AI models of integrated processes of the brain, both here in the neuroscience community and in the AI neuroscience community. And lastly, I wanted to take a second to make a few acknowledgements. One, the CNeuromod team, as you probably saw by the list of authors, it takes a village to run a project this size. The subjects that show up week after week to be scanned is quite impressive. The scanning team that do one-to-one with each of the participants. And of course, La Fondation Courtois, which gives us the money to be able to do this project. Thank you.
Thank you so much. Fantastic talk. So we've got a question of, do you have any health and behavioral data over time in the project?
We've really limited the amount of health data. We definitely have some behavioral data that we're going to start acquiring, but you can imagine with 6 subjects, in terms of the capacity to identify, we've really tried to stay away from anything that would, I guess, inhibit or emerge on their rights, I guess. So we really tried to stay away from, we have a very general health questionnaire, but we didn't go into specifics, nor do we have any neuropsychology. On purpose, we haven't done a neuropsych assessment.
And then there's another question that's, I guess, getting a bit more to what things do you have in place if things go wrong? Like if your scanner breaks down or you have to do updates or, you know.
Like you shut down for 6 months during COVID?
Yeah, exactly.
So Pierre Belec, who's the PI of the project, is also the director of the neuroimaging unit. Scanner breaking down, COVID was a lot of stress, but we work quite intimately with the neuroimaging unit. I think the largest thing that goes wrong in a project like this is actually just availability of the subjects. Life happens. I think otherwise we have everything pretty much under control. And we were really able to catch up. We shut down for 6 months, but then we really intensively scanned. We were the first team back in the scanner, so. And
one final one. How much data is this? Like, do I need, you know, a huge repository to be able to download your data, or?
It's in the, it depends if you want the whole data set or not. For sure, it's a large amount of data sets, but you can access, we have a set of derivatives you can access, but definitely you're looking in the terabytes if you want to access the whole data set. Yeah.
Fantastic. Thank you once again. So we've been informed that unfortunately the first speaker wasn't able to make it today, but we do have a video of their presentation instead that we are hoping to show. Fantastic, yes.
I work as a data steward in the Institute of Neuroscience and Medicine at the Research Center Jülich in Germany. Over the last 3 years, I've had the chance to work with numerous large-scale data sets across many different data providers. And I would like to share some of these successes, challenges, and lessons that we've learned along the way. There are 2 main things that I'd like to talk about. First, the external requirements, such as data use agreements, and the high variability of those requirements. And then second, the internal path that we've taken to coordinate and manage this data to make our lives easier and to work towards fair ideals. Okay, let's get into the external requirements. And the entry point is almost always getting access to the data. Some data sets are open access, and in this case, things are wonderfully simple. The data is immediately available, and it's easy. But other data come with restrictions, and the requirements to meet those restrictions can be significant. Can be anything from requirements for IRB approval, or an up-to-date list of researchers who access the data, which gets to be costly from a time standpoint with regular student turnover. Sometimes we need to declare the research scope, which also gets to be complex when you need to converge many projects into one. And then there are, of course, reporting requirements for annual reports, research progress, etc. And the thing is, I don't think I've come across any 2 data providers who have done it the same way. Pretty much everyone has a different idea for the hoops they want you to jump through to access their data. And I understand why they do this. Neuroimaging data is sensitive data. It needs to be protected. And especially with the enactment of GDPR, we need to make sure that we do this right. But variability is the problem. And the cost of these intersecting and highly variable requirements is a challenge. Because each provider has a different view of what these regulations mean to them, it's not as simple as just complying. We're put in the position of trying to interpret and understand both the letter and the spirit of the rules. and it's challenging to do the right thing when implementing it all on the same workflows and infrastructure. Here's an example of how different data use agreements state the extent to which you can or cannot share their data. This, of course, is an expected thing to cover in a data use agreement, but what I want to point out is the lack of a standard language. They're all generally trying to go over and say the same thing, but there's a decent amount of variability and also terms that are not properly defined, like research staff. That could be something that's very broad or quite narrow. The policy should be clear and not require someone to guess what's meant. And then we get into the security requirements, which are probably among the worst parts of this process. The idea of a security policy is good, but I have yet to read a good one and have found very few that are helpful. Many are neutral or ineffective, and some are just plain counterproductive or insecure. And this isn't a good standard to have or to follow. Security is the overarching objective and point, not compliance for the sake of compliance. And here is the distribution of distinct data policies from data providers we work with. The data providers are on the left and the policies on the bottom. And we see just how little overlap there is here. There are, of course, commonalities, but as I just demonstrated, even when they are addressing a common topic, there's quite the variance in language and expectation. Ideally, we would see more overlaps with vertical columns, but in general, they are addressing different objectives. So now let's move on to the internal approach that we take. And it all centers around coordination. We use a GitLab instance to track all issues and requests, and DataLad to manage our data. The combination allows us to flexibly track and structure the data, facilitate reproducibility, and support collaboration and discoverability. And it all starts with the data request issue. Someone in our institute identifies a data set that they're interested in. They fill out a template and provide as much information as they can, and then others get involved from there. We figure out what does upstream offer, what do people in the institute want to have access to, what are upstream's requirements for us to access their data. We collect all that, submit it, everything's good. Now we have access, and if data is available for download via well-known methods such as S3, everything is simple. DataLad supports this natively. But if upstream requires a custom downloader or some crazy non-scriptable way to get the data, then things, of course, are more difficult. But thankfully, DataLad is flexible and extendable, so we can do it. It's just a matter of the effort cost. A quick example of how we've done this before is in the creation of the DataLad UK Biobank extension. We built it to equip DataLad with a set of commands that allow us to obtain, monitor, and restructure imaging data from the UK Biobank. We are able to track any additions, redactions, or fixes, and also directly restructure the data into a BIDS layout. So then once we have the data downloaded, we clean it up. Sometimes upstream is in great condition, the data is organized according to BIDS, there's sufficient metadata, and everything is consistent. But there are many who do not meet that, and so we have to dig in, discover the problems, fix them, extract info from publications, and so on. It all takes effort, but eventually we get the data into a usable enough state. Then, depending on the dataset, we modularize the content into multiple subdatasets. Commonly, we do this on a per-subject basis or across acquisitions. This gives us additional flexibility for permissions and storage, and for large datasets, it also provides significant performance advantages as only a subset of the dataset availability needs to be acquired. The dataset is then registered into an institute superdataset that acts as the main entry point for all data holdings. The superdataset is hosted on GitLab, improving discoverability, but only exposing the top-level directories. The data and availability information are stored on institute infrastructure where access is encrypted, password protected, and logged to ensure compliance with data use agreements. Credentials are preceded on the infrastructure, making authentication transparent to users. But coordination extends beyond just the initial acquisition of an upstream dataset. It continues through the full data lifecycle. For sufficiently large datasets, which is more and more common, we need to coordinate the computation and storage of derivatives. It's simply too costly for dozens of people to compute and store with no coordination. So we set up a discovery and selection process where people declare their needs and discuss the trade-offs. The resulting derivatives are computed from the inputs, registering their provenance, and then made available on institute infrastructure. Just to highlight, the fact that this resulting data is linked to inputs provides two critical advantages. First, the inheritance of data restrictions can be more easily tracked, enforced, and audited. And second, the provenance capture enables the deletion of the derivatives when they're no longer as valuable to people, because we can just reliably compute them again if needed. This greatly reduces storage demands and keeps our IT team happy. Then finally, upon a completion of a project, an issue is opened to archive it. Only the few relevant bits are kept, so important metadata details, how the results came to be, and then the data with linked publication. Then this is all pushed into a secure, low-cost archive. And that's a quick run-through of our system. If you're interested in learning more about our approach and seeing the full picture, I encourage you to check out my poster, which is available online at this link. Thank you for listening.
Fantastic. With that, we'll bring the session to a close. Thank you from us for attending. Do go and visit the speakers' posters where you can ask all of those remaining questions. And yes, please can we just thank the speakers one last time for their fantastic talks.